Hello everyone, so today our video will be about our primers. The idea of our primers revolves around the rather recent uproar of the myth that soybean milk and soy products cause cancer. On one hand, some have argued that a specific protein present in soybeans, called isoflavones, lead to an increased risk in developing breast cancer. On another hand, optimists have argued that isoflavones lead to a decreased risk in developing breast cancer. As a result, we have decided to delve into this topic to investigate if isoflavones even affect the risk of developing cancer. So, how we will go about this experiment is that we will first identify the gene that expresses isoflavone. Next, we will amplify this gene using PCR, in which primers specific to the gene of interest will be created to aid the PCR reaction. Moving on, transfection will be carried out to insert the amplified gene into a test subject, which is a rat, through either the use of lipid-based reagents or electroporation. With the presence of a control test subject, the control and experimental subject, which is R2, will be observed over a period of time to evaluate the presence of any exhibitions of behavioral or biological changes which either show an increased or decreased risk of developing breast cancer.
Before creating primers, we had to look at the criteria to achieve good primers. Firstly, optimal primer length must be between 18 to 22 base pairs, as it is long enough for adequate specificity but short enough to bind to template strand. Next, primer melting temperature, TM, should be around 52 to 58 degrees Celsius. This produces the best results. Primer annealing temperature, TA, should not be too high as primers will not be able to bind to DNA template, but also should not be too low to avoid mismatches. GC content of primers must be 40 to 60%, and G and C bases have to be present within the last 5 bases from the 3' end of primers. This promotes specific binding. Additionally, there should be no primer secondary structures, and the primers should not contain repeats, which are dinucleotides occurring many times consecutively, as they can misprime. Primers with long runs of a single base should generally be avoided, as they can also misprime. Finally, template secondary structure need to be avoided. If primers are designed on a secondary structure which is stable, even above the annealing temperatures, the primers are unable to bind to the template, and the yield of PCR product is significantly affected.
Using GeneRunner, we ran an analysis on the isoflavone synthase 1 gene from the National Center for Biotechnology Information website (NCBI) to find the open reading frame, or the ORF. The longest open reading frame, frame 2, was used. With the open reading frame, the protein coding sequence was identified. We created primers according to the start and stop codon of the ORF, and that an optimal primer length is about 20 bases. For the forward primer, we listed all 18 possible primers in the 5' to 3' direction that included the start codon ATG at the start to the end of the sequence by moving down by 1 base. For the reverse primer, all 18 possible primers in the 5' to 3' direction that is complementary to the given gene sequence were listed. These included the sequence complementary to the stop codon TTA at the start to the end of the sequence by moving up by 1 base. An analysis using GeneRunner was done to find out each primer's properties, and these were tabulated into a table.
Alright, so as mentioned earlier on, once we have all possible forward and reverse primers, each consisting of 18 primers, we then use GeneRunner software to examine the quality of our primers with respect to the criteria that have been mentioned earlier. For the TM row labeled in red, it means that it doesn't fall within the ideal range. For the secondary structure, also written as SS, those that are highlighted in red, it means that it does not contain any secondary structure. And for the GCC, highlighted in red means there is an absence of GC clamp, and for row labeled repeats and runs, labeled in red, it shows the absence of repeats and runs respectively. For the first round of selection, we look at the primers that have the color green for TM, red for SS, green for GCC, and red for both repeats and runs. For the forward primer, there is only one primer that fits our selection criteria, and that is the primer at number 7. It has a TM of 57.4 degrees Celsius with no secondary structure and has a GC percentage of 50%. As for the reverse primers, there are two possible primers. They are primer at number 5 and number 10. Both of them have the GC percentage at 40%, and both do not have runs and repeats. The difference between primer number 5 and primer number 10 is that primer number 5 has a higher TM at 52.6, while the primer at number 10 has a TM at 52.5 with a difference of 0.1 degrees Celsius. Hence, we decided to choose primer number 5.
In conclusion, our primers are designed and chosen by taking into account of key criteria such as primer length, melting temperature, annealing temperature, GC content, runs, repeats, and finally secondary structures. With this design primer, we are hence then able to amplify our gene of interest that expresses isoflavones using PCR and transfect the product of about 1020 base pair long into an experimental mice and compare it with a control mice to observe any differences in their behavior and biological change to allow us to conclude if high isoflavone levels increases or decreases the risk of developing breast cancer.